Actually, there's so that's fine.
All right. Well, thanks everyone for joining us today. If you don't know me, my name's Daniel Teresi. I'm the Research Innovation and Plant Protection Lead here at Reno's Much Move Forward. First of all, just some little housekeeping notes. If you want to ask questions, maybe we'll save all the questions to the end. We'll have some space, maybe 5-10 minutes at the end. If we go over time, we won't take it personally if you want to leave. If you have other commitments, but you're welcome to stay to further the conversation. But for now, I'm going to introduce Steve Peters, the chair of our board of directors.
Thanks, Daniel. Hi, Steve Peters, chairman of OGVG, on behalf of Richard Lee, our executive director, who's in Washington right now, trying to do some advocacy work on behalf of border issues and the whole board of directors. I just want to say thanks very much for coming out today for the lunch and learn. It's important to us at OGVG that we engage with you, the growers and the team of, who supports the growers. And this is one of many events that we continue to host, and we're happy to do that on your behalf. I just, on behalf of the board, just want to thank Daniel and welcome Kara and Rose. We appreciate Ag and Ag Food Canada and OMAFA for their interest in this and being part of this. So anyway, I hope it's a good learning exercise. And as I say, on behalf of the board of directors, thank you very much for attending. Back to you, Daniel.
All right, so this topic today actually came about a couple weeks ago. So as we all probably know, there have been a couple of sightings and some confirmed cases of pepper weevil in the Leamington, Kingsville, Essex area. So the last time we saw a huge outbreak of pepper weevil was quite a few years ago. So since then, quite a bit has management strategies have been updated and changed a little bit. So to account for that, we planned this refresher course. So I'd like to introduce, if you don't know them already, Rose LeVay, entomologist at Agriculture and Ag Food Canada, and Kara McCurry, who's the IPM greenhouse IPM specialist at OMAFA. So thank you.
Daniel. Thanks, Steve. We also really appreciate OGVG for this awesome facility. This is a very comfortable space for us. We tend to do a lot of our information sharing here. And also thank you for providing lunch for everybody. That's a nice little bonus, I think, that encourages extra participation. So yeah, Rose and I, we like to do these joint talks because we work so closely together on arthropod pests in Ontario. So we're going to kind of jump back and forth through this presentation. And really this is a refresher for those of you who lived through pepper weevil a decade ago. And for anyone who's new, this might be new information for you, but we did try to update it because for the better part of a decade, we actually were working on pepper weevil locally. So this is an opportunity to share some information that we haven't actually been able to share before. So this is just a brief outline. We're going to talk a little bit about the distribution in North America, biology and crop damage, some reminders about, things to look for. Also, we're going to talk a bit about the risk of pepper weevil establishment. I think this is a timely conversation for this time of the year, and especially in light of the recent detections. Then we'll walk through some of the IPM strategies with some new information, some things that we learned from the work that's been done in Harrow and also collaborating with growers in Ontario. And then we're going to talk a little bit about next steps. We have some ideas, but mostly we want to hear from you, what you want from us next. So as far as the distribution in North America, pepper weevil is native to Mexico. So in Mexico, they have year-round pepper production. There's fields, there's greenhouses, there's no real winter. So pepper weevil just exists year-round and jumps from crops to crops. It's a little bit different throughout the rest of North America, so the US and Canada. Pepper weevil has established in some of the southern United States. It's not regulated anywhere in North America except for New Mexico. Some regulations about, I think it's more how to clean up your fields. That state gets a little bit involved with those types of things. But the southern part of the US has a climate that is more conducive to pepper weevil surviving year round. Once you get north, so more northern states in the US and also Canada, we have this thing called winter that pepper weevil doesn't really like. So it can't survive outdoors, but it can survive in a greenhouse. So we do get introductions every year, pretty much every year. And there's a number of ways that it gets introduced into Ontario. So it can get introduced on imports. So imported fruit, especially from Mexico, could be from other states that in the US that have pepper weevil, but most of it is coming from Mexico. They have year-round populations. And so they are always dealing with pepper weevil. So we can import it from there. They are also very good hitchhikers. So there's a lot of records, some good documentation in the US about how even the pepper weevil populations that exist in the southern parts of the US, they migrate north in the US every year and they just hitchhike on vehicles and equipment, and other sort of logistical channels, even people. And we did actually see that here as well. So they can get around. They can also locally be moved through wind events. They're not very strong flyers, but they still get around a number of ways. So if we look at the timeline in Ontario or in Canada, the first documented introduction into Canada was actually in BC in the 90s. There was a fair amount of work that was done at that time. So we've learned a lot, about management from that situation. In Ontario, the first documented case was in 2009. If I go back to my predecessor's records, there were some suspicious cases, potentially a little bit prior to that. But that's, generally accepted as our first introduction was around 2009. So then we jump ahead to 2016. So we call this the year of the weevil because that was our big outbreak year. So Rose and I both started in our positions in 2015. So I was January, you were June, July, yeah. And so when we look back to 2015, there were a few rumors and a couple of reports throughout the summer, mostly towards the end, maybe into the fall, of pepper weevil around. So we know that pepper weevil was in town, like in Essex County, in 2015. We didn't really know the extent of it. So we have some theories about what happened. We think that there was a decent population into the end of 2015, that some of them survived the winter. We had a very mild winter that year. And so we had an already established population in the greenhouses in 2016, because we had a pretty explosive population growth through 2016. So it wasn't really until sort of like mid to late summer in 2016 that we realized it had spread outside of Essex County. It had spread to all the field pepper growing regions in multiple counties. And so we started to take a closer look at what was happening beyond sort of the Essex County pepper greenhouses. And we started working more with field growers as well. And so every year we're gonna include a bit more information about what we've been doing each year since then, but there's always pepper weevil that comes up every year. We just, we had a couple of really bad years and then it sort of went away, but not really. And then on this 10-year anniversary of our big outbreak year, we've had a couple of recent detections. So we just figured this would be a good opportunity to sort of refresh our memories and update some of the information. So when we talk about recent reports, so Rose and I, every single year, we get weevils to ID. So they come from suppliers, they come from greenhouses. We work closely with a lot of the greenhouses themselves, but sometimes we get them from consultants. We just get weevils dropped off sometimes to OGVG. And so we are looking at weevils every single year. There are a lot of weevils that are not pepper weevil, that are not of concern, but we do often, at least once a year, get a positive. I think this year is a little bit different, so we're gonna talk a bit more about that in a moment. But one thing that we did, we thought, let's look at iNaturalist to see what's going on there. And what we see are many, many reports from citizens who have purchased peppers from greenhouses, or sorry, not from greenhouses, from grocery stores, who cut their peppers open and then they find a weevil. And they take a picture and they put it up on iNaturalist. So we see this in the US. So it happens a lot in the US too. This, I think, was from Ohio. This screenshot here, this was from somewhere in Canada. It says a product of Canada. But I think the point is we know that there are pepper weevils throughout North America all the time in production, in some situations, but they are frequently found in grocery stores. So there's always some level of risk there when we have imported fruit that has pepper weevil in it, or if we have established populations that are moving around. There's always some level of risk of it getting into a greenhouse. So this is what we're trying to intercept, but to not be super alarmist with these detections, we just have an opportunity to really focus on trying to get ahead of it and not having the same thing happen that we saw in 2016. So one of the things that we did after we realized how widespread it was, we initiated a pepper weevil surveillance in Ontario. So this information really fed into a lot of other things that we did, some of which we'll talk about in other sections throughout this talk. But it also served as a potential early warning system. So we worked with a lot of growers, so greenhouse and field pepper growers, to set up traps. We check them weekly. Our intention is never to out a specific farm, but really to give like a warning sign to, whether we separate Leamington into quadrants or whatever, like to say, it's in the northern part of Leamington, or it's moved into Chatham-Kent, so it's really just meant to like give people some knowledge that they should maybe be looking more closely at, setting up traps and things like that. But it also gave us a chance to learn a lot more about the pepper weevil in Ontario and what was happening with these populations. So another thing that I want to note, so I have the number of sites. We had quite a few sites in the first several years. This 2019, we had 30 sites in 5 different counties, and that was the one year that we never caught a single pepper weevil. We did continue it for a couple more years with fewer sites, and we still had very, few positive sites relative to the first few years. So it kind of trickled out, didn't completely disappear, but it was improving the situation.
Yeah, all right. Thanks, Kara. All right, I'm going to take it from here just to talk about some of the basic biology or some of the things that we've kind of come across in terms of the populations of pepper weevil that have been in here and intercepted and presented or established for some time back in 2016 now. Obviously, we haven't seen it really in the numbers that we have since 2016. But this is a kind of a map I wanted to show. This is an analysis of the genetics of the different populations of pepper weevil from across North America, as well as the sightings from Europe. So there was obviously an invasion within, it was detected in Italy, for instance. I think back, I want to say 2006, something like that. Anyways, I'm not exactly sure the date, but we were able to look at the genetics of those populations. So there's a lot. Yeah. Sorry. And what's interesting, okay, so this is us in Southern Ontario. And if we look at the genetics and we're looking at the CO1 gene, which is what everybody who does work with insects uses as a reference gene for studying the similarities between individuals within a single population or species. And we can actually look at the relatedness of the individuals within the population. This shows that, okay, so in total we analyzed over 160 individuals from Mexico, 70 from the United States, 86 from Canada, Dominican Republic, 45, the Netherlands, 32, and Italy. And of the individuals within Southern Ontario, we actually found only 3 haplotypes. So this means that of a total of 40 haplotypes that were present in all the populations we analyzed from all these parts of the world, we had a very, very small establishment, which means that, you know, there was a huge genetic bottleneck. A single founder population was able to establish within the greenhouses in Southern Ontario in 2016. So you can see, for instance, the major haplotype we had was H22, and actually we can trace it back to a very particular region in Mexico. So this particular variant was not detected in the United States. So it can also tell us a little bit about where the pepper weevil might be coming from that are establishing within our greenhouses. This kind of analysis can also be useful for telling us about resistance of certain populations over others. For instance, we know there's a certain population of pepper weevil within the Yucatan Peninsula that is particularly is able to survive strikingly in very spicy peppers. Like, you know, all peppers that are spicy have a certain Scoville unit, while habanero has one of the higher amounts. And this was a unique population that could survive an astoundingly high level of Scoville unit. So this tells us a little about the local adaptation and maybe how we can approach management of pepper weevil, depending on, you know, what their genetics tells us about the populations, even tells us about resistance of pesticides. So this could be quite an interesting tool for us, and hopefully we'll get that individual that we collected so far this year barcoded to see what it comes to, and if it's the same haplotype as well. I doubt it. Okay, life cycle of the pepper weevil. So obviously, Karen talked a lot about the survey work that was done. This is a refresher for many of you. I know you guys are super familiar with this, so I don't want to dwell a little bit on it, but obviously there's some unique properties of pepper weevil that make it such a great pest. It's a great evader. What I mean by that is, okay, first of all, the female starts to the cycle. She finds a suitable pepper, oftentimes the pepper bud. They really love the pepper bud, but also the pepper fruit. And she'll go in with her long proboscis, and then she will basically carve out a hole within the fruit, layer her egg within the pericarp of the pepper, and that egg, instead of, you know, hatching and then coming out like thrips do, for instance, if surprisingly, it does the opposite. It goes in the middle of the pepper. So this is what makes pepper weevil so difficult for us to manage, because it's super cryptic. It's protecting itself from exposure to parasitoids, predators, and to pesticides. So this is a very unique kind of biology of the pest, and why, you know, and conventionally pepper pests of greenhouse crops, they're so much more easy for us to target. But this one is really challenging, because all of these larval instars develop in about 2 weeks within the pepper. On average, obviously, depending on the temperature, occurs within the protected confines of the fruit. So this creates a big problem for us. In addition to this, there's a few of the life history parameters of the female that are of concern. For instance, the female itself lays between 5 and 7 eggs per day. And then but she can go ahead and lay up to, you know, 341 eggs in her lifetime, and up to, so in a max of 600. So the average is 341. So that's a lot of eggs. Not only that, but these individuals live a very long period of time. So obviously lifespan of the adult is about, so on average it's 80 days, but it can live up to 135 days. So several months of time where it can survive depending on the environmental conditions. The cooler it is, obviously it lives a lot longer. Their development is and their metabolism is going to be a lot faster at higher temperatures. So that kind of compresses the life cycle a little bit. So very interesting biology. And obviously these are things a lot of folks here have already seen, but just a little refresher. It's good. This has been a good refresher for Kara and I as well. So this is the exit hole that the adult makes. It's significantly larger than that little puncture hole that the female lays her egg in. So the adult emerges from the bud or the fruit. This is indicated by the clean and round hole that displays the presence of the emerged adult. Not only do we want to look at the fruit for oviposition and these emergence holes, but we also want to look at sometimes the foliage. We've seen perfectly well that these pepper weevils, they don't prefer it, but they absolutely can lay and develop completely within the stems of pepper plants as well. Here's an example of the larvae within the pepper fruit, and you can see this one is heavily infested. Another very interesting note about the biology is the female that she puts a plug after laying the egg, and this plug, it actually serves as a deterrent for other females, so that they find their own individual pepper. So it's kind of it helps to disperse the species so that not all of the larvae are developing within a fruit, which increases the risk for the loss of the fruit and obviously the loss of the larvae as well. Okay, so talking about the prevalence of pepper weevil within North America, you can see the parts in red here are the places where pepper weevil is almost always present every single year. And then the parts in yellow are the places where it's seasonally present. It kind of ebbs and flows depending on the seasonal conditions. So you can see the parts that where pepper weevil is always around, places like Florida, California, Texas, etc., and of course all of Mexico as well. But for us, we basically are lucky. If we can get rid of it in the winter, winter certainly serves as a good barrier for the species. In Mexico, obviously this data is really outdated back to 2016, but losses annually of between $30 and $50 million a year. For the United States, about 23 million a year, and I actually think this is really too low of a value to this. Today, we would see a much greater loss due to pepper weevil. In Ontario, our losses due to the occurrence of the pepper weevil in 2016 alone were about $67 million as an estimate. So a very significant pest of concern. Looking at another very interesting aspect of the biology, some work done by Dr. Catalina Fernandez, who was a PhD student in my lab for several years during the pepper weevil outbreak. She studied the prevalence of the ability of pepper weevil to establish in different pepper types. And you can see this is the scale of Scoville units, the spiciness of a pepper. She found that the development of the larvae within the pepper fruit correlates very well with the Scoville units. So you can see the lower the Scoville units, the least spicy peppers develop the best populations of pepper weevil, whereas things like the habanero pepper are some of the peppers that are most resilient. So certainly capsaicin, which is one of the molecules produced by peppers, is a deterrent to the successful development of the pepper weevil, which is kind of interesting. Very important from a management approach is that pepper weevil can very much develop in many solanaceous hosts, weedy hosts, such as climbing nightshade and eastern black nightshade as well. So be aware that these species are prevalent in our area outside, and so they serve as alternate hosts. So being aware of clearing the spots around greenhouses from these hosts is very important as well. And just a little note about the cold tolerance and the overwintering the species is obviously we know pepper weevil is not going to survive a Canadian winter, and this data is supported by the work that Catalina did as well. This shows that the survival of adults is very poor over time at 0 degrees Celsius. In fact, they could only survive about 300 hours before they're all dead. So that's about 12.5 days. So less than 2 weeks exposure of all adults to 0 degrees results in total mortality. So that's very interesting. And of course, there's also other data, very old, from Dave Gillespie and Costello back when they had pepper weevil in British Columbia. It shows that within, whether we have food available for the pepper weevil or not is a huge factor in their survival, not surprisingly. So you can see this is the LT50, the time to mortality for the pepper weevil. So you can see they live a long time, over 100 days in the presence of food at many different temperatures ranging from 2 degrees to 27. But on the right-hand side is without food, and you can see survival is much reduced. The lifespans are much reduced with a total mortality of the population. You can see the best mortality or shortest lifespan is 7 days, or actually it's actually 4 days. Sorry, 7 days for total mortality at 27 degrees. So based on this information, there was basically a recommendation that if you wanted to make sure your greenhouse is free of pepper weevil, you leave your crop-free period to be about 10 days at about 25 degrees Celsius. So that's a general recommendation, but obviously, greenhouses are never constant temperature. So factor in what is the typical condition now to either increase the amount of time or decrease it depending on the temperature. So that's where we're at. With that, I'm going to pass it on to Kara here for this part.
Another thing that I wanted to add, so we don't have a slide on this, but I think it is sort of a good follow-up to that previous slide talking about that crop-free period. So when we're looking at traditional pepper crop cycles, so not under lights, where the cleanouts are happening in the fall, that's where those strategies are really important. And I know it's like a really hard sell to ask a pepper grower to keep their farms while they're cleaning out and not earning any income from selling peppers. That's a big cost, but in the case of pepper weevil, that can be an important strategy for cleanout, because they do survive cleanouts, and we definitely have seen that over the years. The other point that I wanted to make about that, we did in 2016-17, we did some winter surveillance, and we were really targeting, everybody was sending all of their waste to the landfill. There was also some waste going to ETI. So if anyone's familiar with them, I don't think they're in existence anymore, but it's another area. I think we're in Cotton. So we looked at those we looked at 2 different landfills and ETI where waste was going, and we set up traps around those areas, also in the direction of prevailing winds up to like 10 kilometers away. So we did, in some cases, 5 kilometers away from the landfill, nowhere near any pepper farms, get large numbers of pepper weevil on those traps into November even. So we think they were probably carried by the wind in that case. But past mid to late November, we never caught a single weevil. So it kind of supports this, there's still some risk when we're trying to dispose of plant material, but it's reduced, once the temperatures get low enough. Still something we are always trying to figure out what to do with greenhouse waste. But so this is pepper weevil, the risk of pepper weevil establishment heat map that we put together that can roughly give you an idea of where the biggest risks lie. And I just want to say this is going off the assumption that you did not have pepper weevil last year. So you likely don't have a population that survived the clean out. And you are not packing imported fruit, because in both of those cases, that risk map will change dramatically, and there's an increase of risk throughout the entire year if those 2 variables exist. So what this really means, so we looked at, when are you opening vents? We looked at temperatures outside, roughly, imports coming in from Mexico. Like, we looked at a bunch of variables and then pieced them all together to see where's the biggest risk window. And why do we think that's important? Because actually right now is our critical window for early detection and early establishment. So this is where like the best monitoring needs to happen, because if we get early introductions into production houses, then we will see this happen throughout the rest of the summer, where the population really starts to build. So yeah, that is basically the point, is like, this is our best opportunity to intercept the establishment in a greenhouse, and the movement from farm to farm and county to county. So that was sort of the point of that risk map. So now, we kind of talked a bit about the distribution, some biology, some risks associated with establishment. So now we're just going to walk through some of our ICAM strategies and incorporate some of the new information that we have through a lot of the research that Rose has done in Harrow and other things we've done with farms. So biosecurity is obviously, a common theme for any pest. It's really our first line of defense. We're trying to intercept the pest from entering the greenhouse, entering a production area, and also the movement throughout your crop or to other neighboring areas. So there's quite a few things that can be done that actually kind of fall into different buckets, but a lot of them we can really think about, in a biosecurity program. So if you're importing, you want to be inspecting imports. This made a big difference, I think, with introductions in Ontario was some diligent inspections of imported fruit from pepper weevil regions. Also, the idea of not packing near a production facility remains very important for reducing the likelihood of an infestation into a production greenhouse. Employee hygiene. So we talked a bit about how they're hitchhikers, so they can hitchhike on people, on, vehicles, especially on equipment. So just, being mindful of not going from farm to farm, like one pepper greenhouse that has a pepper weevil outbreak and going to another one. If you are a farmer that has multiple farms, that is something to consider. And then piling culls. So, this is not really news. I think that this is risky behavior for reintroductions. They can also survive on the vines outside as well, but this is where you can still get emergence of developing pepper weevil coming from the fruit. Proper disposals, we talked about that, and then hitchhikers, sealing as much as you can the doors and walls. There was a lot of efforts that went into that after 2016. This picture here is a vehicle, and I took that photo in 2017, I think. And I just noticed this little guy on the car, and then if you zoom in, that's a pepper weevil. So I had a story from someone from a farm. They worked at a farm, they left the farm, and they lived 85 kilometers away. And while they were driving, they saw a pepper weevil on their windshield, and when they got home, the pepper weevil was still on their windshield. So that is just, a good example of how they can move long distances too, when hitchhiking on something like a car, even pretty innocently from someone that just worked at the farm. So, the other thing that we've sort of brought up already is the importance of early detection. That the early detection is really one of the most important things you'll do to be able to stay on top of the populations as they're building. So, monitoring obviously is critical. Using, people are probably your greatest asset. So if they're trained, the people that know what to look for, it takes, people that are scouting specifically for pepper weevil, you're looking for ideally a tiny little dimple in a fruit and a tiny little fruit. So it takes a bit of practice to get there. And, we have some tools available to help. There's some posters. You're free to take them. There's English and Spanish over there that have some photos. But one of the things that we've been using are pheromone traps. So these are aggregation pheromones that are released by male pepper weevil. They are they attract both sexes, so they'll attract both male and female. They are short range, so they are really only attracting pepper weevil that are within 6 to 9 meters of that lure. So there was some concern initially that the lures are drawing them into the pepper greenhouse, but the peppers themselves release so many volatiles, like they are smelly to pepper weevil. So the monoculture of peppers really is what's drawing them in. And then you need enough lures set up in the greenhouse to be able to detect them there. So there's some strategy with how many to use. The more you have, the more likely you are to detect them early. But ultimately, they have to be within 6 to 9 meters of that lure to be drawn to that card. But the aggregation pheromone, it attracts both sexes because it's basically saying this is where the party's at. So there's food and there's mates, like come on over here. So what that means too is that because the males release these pheromones, pepper weevil, like both sexes will join them, that they're usually found in hotspots. So when you first detect one, a lot of the effort should be focused in that area and trying to sort of, quarantine or keep it contained as much as possible. You can put traps inside or outside. So our surveillance work was outside. We also worked closely with farms that were trapping inside and sometimes shared that data with us as well. And then of course, plant inspections, looking for signs of damage are important. So we do have this video that we made that is available to watch online about setting up traps. For outside, these are like sign stakes that we use that seem to work really well. But for anyone that's interested, you can take a look at that video. So the other thing about monitoring though is we get so many weevil. There's so many weevils. So there's hundreds of weevil species in Canada, in Ontario, and there's also, some of them are very obviously different from pepper weevil. Some are very similar. So, we can get what we call bycatches. So they might not, or they're not pepper weevil, and they are also not a pest of a pepper plant. So we're not really concerned about them, but we need to tell them apart. So Rose and I, like I said, we get a lot of weevil samples every year. We're happy to do this. We love to be connected with everybody, but we also want to give you some tools, to minimize maybe the panic when you see a weevil on a trap. So we did write this article in Greenhouse Canada magazine that has some good photos, and I'm going to show you a couple right now, but just as an additional reference, you can go back to. But some of the features of pepper weevil are pretty distinguishing. Their body shape, they have an arched back. They have these really like coarse scales. So like these white hairs can be a little yellowish, but they're sort of haphazard compared with some other weevils, like sort of, we think of like Albert Einstein, it's like crazy hair. They have these spurs on their femurs, this little concentration of scales at the top of their wing covers. There are other species that have similar features though. So this is where things can get sort of interesting. The other thing I think about pepper weevil that might be important, although this is, may require practice and a little bit of training and is not so easy, even for those of us that have looked at these kind of features, but it might be important to know if you found a male or a female. So they are a similar size, the males and females. A lot of insects, like the males will be smaller and the females are much bigger. Sometimes it's really obvious, but for pepper weevil, they're a similar size. And there's a few things that you can look at that can tell you if it's male or female. And why that matters. If you are monitoring closely and you find a single pepper weevil on a trap in your farm and you see no damage and it's a male, then there is much less risk of an established population. You should be alerted to the fact that pepper weevil is nearby, but if it's a female, there's a chance that they've already laid some eggs. And so, that's maybe why this is meaningful. We will also share these slides after if anyone wants to take a closer look at some of these things. So these images came from that Greenhouse Canada article. So part of this was looking at what we found on these traps when we did our surveillance work. And also, probably more importantly, what are the most common other weevils that we get from greenhouses to ID? So these are the ones that seem to really trip people up a lot. And I also understand, even if I see a pepper weevil or I'm certain it's not a pepper weevil, I will always ask Rose to look at it. And then we always ask Rose two technicians. So we have 4 people that are confirming whether it is or it isn't. So I know sometimes we all want that sort of reassurance, but there's a couple of Pseudorhynchus species, a couple of Titius species. They seem to be really common early in the season. So right around now, they're probably emerging from their overwintering sites. They're looking for something to eat. Some of them, you know, they eat clover, they eat cabbage. They're not important for peppers, but we can find them on the tracks. It's rare if you see a weevil on a plant that it would be anything but pepper weevil, but it can happen. It's just rare. This is mostly what we would see like on the sticky traps. This would actually really tripped us up when we got this one because it looks so similar to pepper weevil. It's actually really difficult to tell species. So this is a whole family or subfamily. One of the things that was really interesting about the specimen we got, though, who's a little damaged there, was it was a male, and he had only the males have these prosternal spines that come from underneath their heads. So that was a good indicator right there because pepper weevil does not have that feature. And these fun facts are used in male combat in this species, but definitely an indicator that that was not pepper weevil. The other thing that was really striking about this species was it did not have a strongly arched back. And you can see it's just not as beefy. It's a little bit smaller. The other one that can cause some confusion too are other anthonymous species. So these are in the same genus as pepper weevil. Some are native to Ontario. There's quite a few. There's like 17 anthonymous species native to Ontario or that have been found in Ontario. But they sometimes will have very distinguishing features like the patterns on their hind wings. But when they're stuck to sticky cards, sometimes the colors change, sometimes the scales rub off. It's still sometimes difficult, but that's just another example there. So once you've identified it, you know that you have, you know, pepper weevil on your farm. You want to sort of think about, you know, where to put your efforts. This is just a little snippet from some of the surveillance work we did that really shows where pepper weevil populations start peaking in September and October. So they're building early on, and then the population dramatically, you know, increases a certain time of the year. So this is where like the strategy is really like you want to gain control here. And not wait till here. So you might want to increase inspections, you know, plan for extra labor, because a lot of it comes down to that. Another thing we did was look at exclusion screening. So we're just trying to figure out what is the biggest hole that we can use to exclude pepper weevil. So we tested a bunch of different sizes. We worked with Calling Gas. They made these special for us, and we created these little chambers. You release pepper weevil on one side. They have to go through the mesh to get to a pepper plant with little fruit and flower buds on it. And then we monitor it to see what happens. So the 2 by 2 millimeters, 100% of them made it through. This was really interesting. So because they're kind of beefy little weevils, they couldn't squeeze their bodies through a 1 by 2.5 millimeter hole. So that was a, you know, pretty good starting point. A lot of growers that installed screens or some of the new builds that installed screens went down to 0.8 by 0.8 because the smaller you go, the more you can exclude. So you can exclude other pests as well. The other strategy that is really important is roguing. So when you have scouts that are looking for signs of pepper weevil, you also can have scouts or employees that their job is to remove anything that is suspected to be infested. So these are rough estimates for the number of employees that you might need to assign in these different scenarios. So here we're looking for dimples. So this, I would say, is more of like an early stages of infestation. Once you start seeing this, there is developing larvae and pupae inside those fruit. So you're closer to the point where the adults will be emerging. So 1 employee per acre. And then once you see emergence holds, you know you have a reproducing population, and so you're up to 2 employees per acre. So these numbers kind of came from talking with growers that were implementing a strategy that really focused on the labor aspect of this. It's very labor intensive. We were fortunate enough to have a farm who came to OGVG, gave a talk, shared their data, and really talked about their success story with this. And they used bounties, so they hired extra labor, they paid money for every single weevil, every single pepper, every single, they inspected the peppers at the end of the day to see if they actually were pepper weevil, and each employee got paid bounties for all of the ones that they found. But they showed this graph of how the population went like this and then like that, and a lot of it had to do with labor that they invested. So yeah.
Okay, switching gears to kind of other management approaches. Obviously, Kara was talking a lot about prevention and scouting and things like that. I'm going to cover very quickly because I realize we're at the top of the hour. What we've been working on for management approaches, what can we use in Kara if ever this becomes established again? So things we've been looking at, I'm going to cover SIT, biocontrol. Kara's going to talk about pesticides, conventional microbials and biocontrol pesticides. Okay, so the first one is SIT. This has been in the news recently with you guys have heard that the New World screwworm is creating a huge issue in the southern United States and threatens to jeopardize the whole cattle industry, the meat production industry. And back in the 1950s, when the pest presented itself in huge numbers, they used the sterile insect technique to effectively eliminate the pests completely, which is a really effective strategy. So we were really interested in using the sterile insect technique to try to get rid of the pepper weevil. So we worked very closely with Cynthia Scott Dupree at the University of Guelph, Bruce Power and Nordian, who provided us a facility in which to irradiate pepper weevil at. So basically the student, Jacob Bassett, who's now coming back to Hera very shortly, so we're really happy about that, worked tedious hours to try to extract those pupae, sterilize them at the facility at Ottawa, and then personally ship them there and bring them back to Hera to study their biology. And so basically the idea is that you're flooding the system with completely sterile individuals, and they serve as a sink for the reproduction of the individuals who are not sterile. So in that sense, you can actually flood the system and eliminate within a single generation, oftentimes. It's a very effective strategy if you find the right species. So this is the pupae ready for irradiation. Jacob compared very many doses of irradiation, and you can look at, he's actually managed to find the exact dose of irradiation, 110 gray of gamma radiation completely eliminates the potential for both males and females to reproduce. So you can see here, females are a little more sensitive to irradiation, whereas males are a little bit more resilient. They continue to produce offspring. So that's very interesting. Note, though, that egg sterility was 100% also for at 110 gray. But that one thing we did notice is that irradiation does it shortens the lifespan of these individuals. So you do have to constantly reintroduce individuals to make this strategy work. So that's something that we did learn, and I think it could be an interesting option. There's a little bit more work to show, but we're really compressed for time here. So I'm going to cover biocontrol. So obviously back in 2016, lots of farms, fields, and greenhouses had pepper weevil in them. So basically, we did a lot of collecting of these infested fruit, and amongst them, we found some parasitoid wasps that are attacking the pepper weevil. So the biology, basically, you saw the cycle previously, but so what's happening is that parasitoids will attack, again, sometimes the egg, but in the case of many of the species for pepper weevil, they'll attack the larvae within the pepper weevil. So this is at the stage here. The egg is laid within the fruit. Several days later, the larvae hatch, and then, and for the species that we were really interested in, it's a coinobiologist, a species that attacks the larvae. Okay, back for 2016, this is an example of the data we collected for other surveys we did for the natural enemies of pepper weevil. This shows the spike of the pepper weevil emerging from the many thousands of peppers we collected from multiple different farms, 10 different sites across southern Ontario. And you can see this is a seasonal peak. You see the numbers, obviously, it's right within that window that Kara already suggested. Lots of pepper weevil out in September, October, and November, peaking in November, and then dropping because the temperature is dropping shortly after that. Here's a summary of what we collected. Almost 3,000 peppers collected. Of those, we collected about just about 3,000 pepper weevil as well. A very small number of wasps, but very important. Some farms had up to 10% parasitism by wasps, naturally occurring. That was one greenhouse facility. Thank you so much, folks, who contributed to that, because I think we learned a lot of really interesting stuff. Here's a summary of the species we found. 43 parasitoid wasps in total, 7 different species. So 3 species in Mealeolus, 1 Eupelmus cultriceps, Haliscoa hunteri, Pteromalus autonomy, and Bracon. Here you can see the distribution of the numbers we found. A high bias towards females. Sometimes very few males. There are some exceptions to this, but largely the individual we collected were mostly females. The species we're really interested in is this one here, Haliscoa hunteri, because copper folks, I'm looking at you folks here, happen to have a colony of this species. And because we already found it documented in Canada, we were able to collaborate with Copper to bring it in and to study it. And this work was done by Serena Leo, who was a master's student at the University of Guelph. She studied this wasp very carefully. This wasp is interesting because it reproduces in faster than pepper weevil. However, it's very small and its ovipositor is very short. So reaching those larvae deep down in the pepper, very difficult. It really likes the tiny fruit, tiny buds, but that doesn't matter. It's still important. Here we go. This is a summary of the work she did. She looked at the chemical ecology, the attraction of the wasp to pepper weevil. Specifically, this is infested fruit with different life stages. First instar, so we looked at actually chickpea too, because this is the alternate host that Copper uses for rearing, and we did this too in the lab because it produces thousands and thousands of wasps very effectively in a very confined space. So we can actually mass produce and release these wasps. And then pepper, so infested with first instar larvae versus third instar larvae of pepper weevil, or first instar larvae versus third instar larvae, fourth instar larvae for the cowpea or chickpea weevil, the alternate host. And you can see relative to the clean fruit with no infestation, the wasps were constantly attracted in Y-tube studies. So I'm going through this very quickly, but it's a lot of work. And she showed that they are very much attracted to the volatiles emitted. So basically, this is a Y-tube. In one arm, you put the attractant, the clean pepper versus the infested pepper, and they almost always go to the infested pepper. So it suggests these pepper are emitting a very interesting, unique chemical signature that can be detected. Folks at Nature Fresh use this. They developed a chili, the dog that could smell it, and that was amazing. The dogs can smell it, so can the wasps. And we actually characterize some of the compounds associated with these unique pest incidences. So this kind of information, we're also working with a group at the University of Windsor to try to study, can we develop sensors for pest detection, which I think is a very interesting application. And of course, we did parasitism work. We looked at infesting small pepper fruit, ornamental pepper plants. Would be easily contained in the lab. And we looked at things like exposure period, larval life stage, and also fruit size. As I mentioned, fruit size is important. We found that the best suppression of pepper weevil was in the fruit that were the smallest. So that means the wasp has the easiest time reaching the host within these very small buds and very small pepper fruits. So never forget to check those small buds. It's very important. And whereas the bigger ones have a lower degree of suppression by the wasps. We also did a really lovely trial with the Truly Green. We're so grateful for that collaboration. This was a summary of the work that was done there with Copper as well. So they provided 20,000 to 30,000 Haliscoa hunteri wasps over weekly for a period of 8 weeks. And this is what we collected. So we collected fruit that were infested by pepper weevil back in, I think it was 2018, yeah. So this is the weekly count of the infested pepper fruit. The black is the number of pepper weevil emerged from the pepper fruit in the control versus those with the Hallis white. You can see that weeks 5, 6, and 7 have no pepper weevil that detected from the pepper fruit that we collected. So overall, I think it suggests that if you are willing to put in that wasp over time, you can get a reduction potentially of the number. However, the wasp does not establish well in bell pepper fruit. They are huge. So if you're expecting it to actually recover and you're not willing to put it in weekly, it's going to be a much more. And okay, I'm going to hand it off to Carol. And then quickly.
Thanks, Rose, for speeding up. We should have planned for an hour and a half. Okay, the last little bit we're gonna talk about is just pesticide efficacy. So we also did trials, testing different pesticides. So we are looking for, some assistance with managing here. We do recognize pesticides are not bulletproof solution at all, but we need, an integrated approach. Sometimes you need to pull out the big guns. We also looked at softer products, so things that might be able to be used throughout production that are soft on bios, like microbials. We also considered organic versus conventional. So we looked at 16 different products, some of which were already registered on peppers, some of which weren't, and we screened them in the lab. So there's 2 phases. The lab trials are really for screening what really doesn't work, and then phase 2 is the greenhouse cage trials, where we test it in a somewhat realistic environment. Obviously, we're not going to be releasing pepper weevils in a greenhouse uncontained. So we had to use these cages. And it's this, strategy we use for pests that we don't want to release into the environment. So these are the products that we tested in a lab. I know this is really small, but there's a couple things that I just wanted to point out here. So there's a few products that we got 100% killed in the lab, and that didn't translate to the greenhouse cage trial. So this is where, the 2 phases really comes in handy, because we can weed out some of the products that aren't effective at all in the lab. So they're not included in the greenhouse cage trials, but then the greenhouse cage trials can further, inform us about what it would look like potentially in a commercial crop. And so these are the results from the greenhouse cage trials. So the very interesting thing that happened here, I think, is that our top performer was kaolin clay. So Surround is the product. It pretty much by far outperformed a lot of the other products, but it still only gave us 80% mortality over 3 applications. The other interesting thing, so the second one here is, sorry, is that the second one? Dun, dun, dun. Yes, is thiamethoxam. So that's flagship. And that was sort of our industry standard. So that's what we were comparing things against. It's a neonic. We assumed it would be the highest kill, but it's actually not. And it's also super harsh on bios. So that was interesting. And then we do have a few other products that offered a decent level of suppression. So the final slide, what is actually registered, we have lost diabrom. So for anyone that's familiar with that product, we used to have a label, could be used after last harvest to manage pepper weevil, but that is no longer. We have 4 products that are currently registered. So flagship, Ryman, XRL. Hervanta is a newer product. So we actually secured a priority at the minor use priority setting meeting in March in 2016. And we just got that label. It sometimes takes between 6 to 9 years to get a label from that priority setting meeting. But once we have it, then we're really glad we got the priority that long ago. But those are currently the 4 products that are registered. We do have these other 2 options. They are not labeled for pepper weevil. We were in pursuit of adding pepper weevil to the label. So what I would say is that if you are spraying these for another labeled pest, then you might also see a reduction in pepper weevil populations. And then as far as kaolin clay goes, we have actually since then submitted packages to the PMRA is no longer the PMRA. That's the federal regulatory body. It's the PRD, the Pesticide Regulatory Directorate. So we submitted packages for all greenhouse crops, trying to get a label for this product. It's brand new. We don't have any greenhouse uses on that label. But we're trying to get a label for in production and after last harvest. And so the science review's been completed and we're just waiting. They have to put it out for public consultation because it's a new use site. It's a pretty benign product, really, like it's clay, but it's super effective. So obviously we want to try to get a label for that. But we might have to get creative with how to use it because it does leave this residue of clay on the foliage and on the fruit. So there might be some need to sort of target, before fruit production or post harvest, or, certain areas. Like we wonder if something like the approach with diatomaceous earth that people were using in the year of pepper weevil, we just might have to play around with it, but we need a label first and then we can see where it fits. But it could also be a good cleanup product from what we can tell. So this is just a summary slide that I'm not going to go over because it's all stuff that we covered. One thing I do want to mention, because Rose talked a lot about her work with Paliscoa, is that as far as I know, this is not a product that's regularly carried in Ontario. And probably, as they're living organisms, they need a little bit of lead time to, figure out if they can bring it here, build up populations or something. So just make sure if that's something you're interested in, that you reach out to your covert reps to talk about what that might work. But we did see, that they obviously can be part of the solution for pepper weevil populations. And that's really it. So I also wanted to shout out to Iris Bitterlick. So she is under contract with the Fruit and Vegetable Growers of Canada, Greenhouse Produce Alliance of Canada. She is the National Greenhouse Vegetable Minor Use Manager and the chair of the National Greenhouse Vegetable Minor Use Committee. All that to say is like a lot of what we have in the greenhouse sector, like the products, and she is in the background, like always getting to get solutions in your hand and a variety of them. So she's helped with a lot of that securing minor use priorities. And then Josh, he's our provincial minor use coordinator. So he's the one that we will go to and say like, Josh, please, we need this, and it's an emergency, or so he is the one who often submits all the paperwork from Ontario, and they're kind of in the background and they don't get enough credit. So yeah. So we are hoping to initiate a surveillance program again this year. We can also maybe do some cleanout refreshers in the fall. We have some Weevil ID tools like the Greenhouse Canada article. And for those of you that have a few minutes, we have lenses and microscopes, and we have a whole bunch of pepper weevil samples. And then we have a whole bunch of dishes with other weevils if you want to just kind of look at them before you go. One thing we thought of that we're curious if you guys are interested in doing like a Weevil ID workshop, we do think having reference specimens would be useful for anyone. When you see a weevil, you can compare it with a pepper weevil. Sometimes that helps. So we have a stash. It's dwindling, but we have a stash. If you wanted to do a Weevil ID workshop, we could work through like some other weevils, pepper weevil. We can everyone can make their own, reference samples maybe to bring back to your farm. Anyways, just some ideas from us, but we would also like to hear from you if there are other things you want from us or like how we can help you. And that's all. There's a lot to get through. I don't know.
So yeah, so thanks, Rose and Kara. Feel free to ask questions now. If anyone has any questions, you can raise your hand and ask them now, or you can stick around and ask them directly afterwards if you're a little bit shy in front of larger groups. And then anyone online as well. Kara and Rose have their emails here, so you can email them, you can type your question in the chat and I can ask it. But yeah, like Kara said, feel free to use the microscopes over there and take home some posters, some reference posters.
So this
And-
talk, here, help your talk. STN is English and ST is Spanish.
We have a question online.
Okay.
What is the effect of cal and clay on bios?
Yeah, so I don't think there's been a lot of work done testing kale and clay on bios. So that's something we didn't do, right? And because it's not a greenhouse product, like I don't think there's been much work done elsewhere, but it would be something that we should look at, and I suspect it might not be good on bios, but that's just a suspicion.
But it might also be like how the timing, in the sense like you spray it binds to the foliage, it binds to the fruit, and it affects whatever's out there. But then after, I'm not sure.
Right.
But if you release your bios after, maybe it's less vulnerable.
Yeah,
We just don't know. I don't know.
because it wouldn't be persistent, like chemical, right? Like it's sort of, I mean, some people refer to it as like a bit of a repellent, but it also just kills them. So it could be similar to what like diatomaceous earth does, right? I don't know. Yeah. So it's something we should investigate though, probably, if we get a label. Good question. Anybody else?
What's the pepper we will eat? Food.
So the adults will do a little bit of feeding on the plants. So sometimes you'll see like a little, like some holes in the leaves or on like the peduncle of the pepper fruit. Sometimes you'll see some chew marks. But the immature stages, like the larvae, they're just eating the inside of the fruit. So, and that's, I mean, partly what makes it so difficult is because they're hiding in there.
Yeah, and of course, what we didn't really mention is the premature fruit abortion, like
Yeah.
the larvae get in there, they're chewing, and then it triggers that premature fruit abortion that, your yield is gonna plummet, after that.
Yeah, and I mean, that's a good point too, because cleaning the floors became really important because of all the fruit that fell. And they're developing still on that fallen fruit, and then they still emerge from those peppers, but. Anybody else? Thanks for waiting it out for as long as you did.
And for the future, if anyone else has any topics that you'd like to hear from industry experts, Kara, Rose, or we have other researchers other than even though they do a lot of lunch and learns for us, we have other industry experts that we collaborate with. So if there's anything else as far as clean out, any production type of lunch and learns that you'd like to hear any information from, you can reach out to myself. And yeah, we can organize an event like that. We actually have just one more question online. Can you disclose how many pepper weevil you confirmed you received this year?
So I personally have had 2. Yeah. I mean, I think that it's really hard to know how widespread it is without people talking, so there's a lot of rumors going on right now. So I am aware of a couple farms, but have only personally received 2. So
And like you mentioned, we're looking at setting up another surveillance study this year. So hopefully with that, we might be able to find a couple more and look at the severity. Um, and those were in the Leamington, Essex area.
Mhm.
Yeah.
Yeah, yeah, and I mean, for anyone, like, we don't ever disclose who, like, it's confidential information for any farms that reach out to us directly, confidential. Any suppliers that reach out to us directly, you don't have to tell us the farm it came from. We are just here to like sort of help, and having an idea of how widespread it is sometimes can help inform other things that we're doing. So just like getting the, pesticides moving, sometimes that information helps us like poke people, or like other projects. We've worked directly with farms before on, testing things on the farm and stuff like that. But otherwise, knowing where it is really is just an indicator of like how it's spreading and the risk that it poses to the sector in different counties. That makes sense.
Perfect. Well, thanks again, Karen and Rose. So this has all been recorded. It's going to be available on our grower website for any of the suppliers here. We can email it to you afterwards, as well as the slides as well. So if there's anyone in your organizations that weren't able to come today, or able to join online, then we encourage you to share this presentation with them. And thank you all for coming.
Thank you.